Okay, so we have learned about these glycolipids and glycoprotein before, but means that they had short fragments of the sugar attached to the proteins of lipids. So can you see that? Short fragments of the sugar, either attached to the proteins or attached to the lipids, or phospholipids, for example. Okay, so they have a short carbohydrates or this sugar change protruding out of the cell surface known as glycolipids and glycoprotein. So in this structure, you are required to label which one is the external surface. Okay, so how we know? Because the role of glycolipids and glycoproteins, they are more to the cell recognition. Okay, whether we can recognize the cells or not is based on this. Okay, glycolipids and glycoprotein, most of them. So it means that they must be in the external surface, correct or not? It's something like always say that glycolipid, glycoprotein is like our car plates, right? The number plates of the car. You don't hide them inside your car, right? Because you want to recognize your car. Okay, if let's say I have 10 my V, okay, same color, same design. Now, how you know which one is your car? You will look for these car plates, right? So inside the cells, also the same thing, how we recognize them based on this glycoprotein and glycolipids. So because there are 4 recognitions, means that this one must be external surface. Okay, so explain why you think that that is the external. This one is the external surface. So because of the presence of the glycolipids and glycoproteins. Okay, so due to the presence of the oligosaccharides or short fragments of the sugar of this glycoprotein and glycolipid indicates that the marked region is the external surface of the membrane. Okay. So what are the role of the glycolipids and glycoprotein? So first we know that for cell recognitions, right? Okay, but they do have other functions. First of all, they help to stabilize the membrane structure. So because these short fragments of the carbohydrates, they can interact with water molecule, so making the membrane more stable. Okay, means that less enemy in this case. Okay, so help the to stabilize the membrane structure. The short carbohydrate chain of the glycolipid and glycoprotein form hydrogen bonds. I will tell you guys more hydrogen bonds when enter into the osmosis. So with the water molecules surrounding the cell, help to stabilize the membrane structure. Okay, so this is a very, very important idea, guys. If you have a cup, you put the water, and then you put a layer of the oil, and then you shake. Yeah, you form the oil droplets initially, but toward the end, they cannot mix together. They will go back to 2 layers. Can I see that? So this is very, very important. If your membrane cannot help to maintain, the droplets like cells, the membrane is not stable. What will happen? The membrane will break. Then the cell cannot survive. Are you clear? Clear. So therefore, membrane actually, in this case, because the oil and the water, they cannot mix together because it's not stable, the structure. Why? Because it's not, they don't have the glycolipid and glycoprotein, right? It's oil. They don't have phospholipid as well. But with the phospholipid and also glycoprotein and glycolipids, they enhance or they stabilize the membrane structure, make the membrane, okay, won't be easily break apart. Okay. What else? As we do know that both of them act as the receptor molecule. Okay, the binding of hormones and the neurotransmitter onto the glycoprotein and glycolipids, which act as a receptor, will trigger serial chemical reaction within the cells. Again, we're going to learn this one in the cell signaling, but not so soon. Okay. Next, glycoprotein and glycolipid can also serve as antigens for the cell recognitions. Okay, so guys, my mistake here, glycoprotein as well. Sorry, glycolipid as well. Okay, both of them can also serve as antigen, which are used to allow cell to recognize each other. But which one more important? Glycoprotein. If the question asks for one, always write glycoprotein. If the question gives 2 marks, write 2, then glycoprotein and glycolipids. Are you clear? The first one you must put in must be glycoprotein. Okay, they serve as a surface antigen for us to recognize them. Okay, so with this, I stop the recording.